Hello students, welcome to readmedprep Academy channel. Today in Applications of Biotechnology-Part-Iii, we are going to talk about the molecular diagnostic techniques like the Elisa, PCR and RT-PCR. Early diagnosis of infectious diseases or inherent genetic defects is essential for appropriate treatment. Early detection of the disease is not possible using conventional diagnostic methods like microscopic examinations, serum analysis and urine analysis. These laboratory techniques are indirect and not always specific. Scientists are continuously searching for specific, sensitive and simple diagnostic techniques for diagnosis of diseases. Some of the techniques that are reliable and help in early diagnosis are recombinant DNA technology, polymerase chain reactions called PCR, and enzyme-linked immunosorbent assay called ELISA. Presence of pathogens like virus, bacteria, etc. is detected only when the pathogen produces symptoms in the patient. By the time the symptoms appear, concentration of pathogen becomes very high in the body. However, very low concentration of a bacteria or a virus, even when the symptoms of the disease does not appear, can be detected by amplification of their nucleic acid. Now let us move on to the enzyme-linked immunosorbent assay, ELISA. ELISA is a biochemical procedure discovered by Eva Engvall and Peter Perlmanin in 1971 to detect the presence of specific antibodies or antigens in a sample of the serum, urine, etc. Their images are given below. It is a very important diagnostic tool to determine if a person is HIV positive or negative. ELISA is a tool for determining serum antibody concentrations, such as antibodies produced in a person infected by pathogens such as HIV, and also for detecting the presence of specific antigens and hormones such as human chorionic gonadotropins. During diagnosis, the sample suspected to contain the antigen is immobilized on the surface of an ELISA plate. The antibody specific to this antigen is added and allowed to react with the immobilized antigen. In the image below, you can see a coated well taken with antigen, and the add specific antibody to be measured to this coated antigen well. The antigen antibody is linked to an appropriate enzyme like peroxidase. The unreacted antibody is washed away and the substrate of the enzyme like hydrogen peroxidase is added with certain reagents such as 4-chloronaphthol. Here you can see in the image below, when the specific antibody is added to the antigen coated well, which is washed, and the enzyme is added conjugated with the secondary antibody. The activity of the enzyme yields a colored product indicating the presence of the antigen. The intensity of the color is directly proportional to the amount of the antigen present. ELISA is highly sensitive and can detect antigens in the range of nanograms. In the image below, you can see where in the last image, after washing the test, the substrate is added and the color is measured. Here, to sum up the enzyme-linked immunosorbent assay, first coating is done. Antigen is adsorbed onto the wells in ELISA plate in coating buffer, and then the buffer is removed and the plate is washed. Blocking is done. A buffer containing unrelated protein is used to block free sites in the wells. After the buffer is removed and the wash plate is taken and detection is done, the enzyme conjugated detection antibody binds the antigen, and after the removing of the buffer and the wash plate, then reading is done where the substrate is catalyzed by the enzyme to generate the colored readout. Right side you can see the key of the different things. There are 4 kinds of ELISA, namely direct ELISA, indirect ELISA, sandwich ELISA and competitive ELISA. The types of ELISA you can see: the direct ELISA where the antigen and the primary antibody conjugate is present and a substrate is added. In the indirect ELISA, the primary antibody conjugate with the antigen is taken and the secondary antibody conjugate is added and a substrate is added. In a sandwich ELISA, where the capture antibody binds to the antigen primary antibody conjugate and a secondary antibody conjugate is added with the substrate. In a competitive ELISA, the inhibitor antigen is added to the antigen and the primary antibody conjugate and a substrate is added. It is a highly sensitive and specific method used for diagnosis. ELISA possesses the added advantages of not requiring radioisotopes or a radiation counting apparatus. Now let us move on to the polymerase chain reaction, shortly called PCR. The polymerase chain reaction PCR is an in vitro amplification technique used for synthesizing multiple identical copies in billions of DNA of interest. The technique was developed by Kary Mullis in the year 1983 and he became a Nobel laureate in 1993. Three steps involved in PCR: denaturation, renaturation of primer annealing, and synthesis of primer extension. The double-stranded DNA of interest is denatured to separate into two individual strands by high temperature. This is called denaturation. Below image you can see the template DNA is denatured and the two strands are separated by denaturation. Each strand is allowed to hybridize with the primer. Renaturation of primer annealing. The primer template is used to synthesize DNA by using TAQ DNA polymerase. During denaturation, the reaction mixture is heated to 95 degrees centigrade for a short time to denature the target DNA into single strands that will act as a template for DNA synthesis. Annealing is done by rapid cooling of the mixture, allowing the primers to bind to the sequences on each of the two strands flanking the target DNA. During primer extension or synthesis, the temperature of the mixture is increased to 72 to 75 degrees centigrade for a sufficient period of time to allow TAQ DNA polymerase to extend each primer by copying the single-stranded template. At the end of the incubation, both single template strands will be made partially double-stranded. Here you can see the temperature control in PCR. In denaturing, the heat is used up till a temperature of 95 degrees centigrade where the DNA strands will be separated. In annealing, 55 degrees centigrade is used where the primers bind to the template DNA strands. During extension, 72 degrees centigrade is used and the TAQ polymerase synthesizes the new DNA strands. A single strand becomes two strands become four strands become eight strands become 16 strands, and the process continues till billions of DNA are formed. The new strand of each double-stranded DNA extends to a variable distance downstream. These steps are repeated again and again to generate multiple forms of the desired DNA. This process is also called DNA amplification. The PCR technique can also be used for amplifications of RNA, in which case it is referred to as reverse transcription PCR, otherwise shortly called RT-PCR. In this process, the RNA molecules, the mRNA, can be converted to complementary DNA by the enzyme reverse transcriptase. And the complementary DNA then serves as a template for PCR. Steps involved in PCR, where the original DNA is taken and denaturation is done, where the separation of the strands is done. Then the next step, annealing, is done, and the third step, extension of the strands, is done. How RT-PCR works? Here the RNA strand is taken and a complementary DNA strand is synthesized by the enzyme reverse transcriptase, and the complementary DNA RNA hybrid formation occurs and the DNA amplification is done after the complementary DNA is formed. PCR in clinical diagnosis. The specificity and sensitivity of PCR is useful for the diagnosis of inherited disorders, genetic diseases, viral diseases, bacterial diseases, etc. So, the diagnosis and treatment of a particular disease often requires identifying a particular pathogen. Traditional methods of identification involve culturing these organisms from clinical specimens and performing metabolic and other tests to identify them. The concept behind PCR-based diagnosis of infectious diseases is simple. If the pathogen is present in a clinical specimen, its DNA will also be present. Its DNA has unique sequences that can be detected by PCR, often using the clinical specimen, for example, blood, stool, spinal fluid, or sputum in the PCR mixture. PCR is also employed in the prenatal diagnosis of inherited diseases by using chorionic villi samples or cells from amniocentesis. Diseases like sickle cell anemia, beta thalassemia, fetal ketonuria can also be detected by PCR in these samples. Complementary DNA from PCR is a valuable tool for the diagnosis and monitoring retroviral infections. Example, tuberculosis by Mycobacterium tuberculosis. Several virally induced cancers like cervical cancer caused by papillomavirus can be detected by PCR. Sex of human beings and livestock, embryos fertilized in vitro, can be determined by PCR by using primers and DNA probes specific for sex chromosomes. PCR technique is also used to detect sex-linked disorders in fertilized embryos. Now, what are the applications of PCR? The differences in the genomes of two different organisms can be studied by PCR. PCR is very important in the study of evolutions, more specifically phylogenetics, as a technique which can amplify even minute quantities of DNA from any source like hair, mummified tissues, bones, or any fossilized materials. PCR technique can also be used in the field of forensic medicine. A single molecule of DNA from blood stains, hair, semen of an individual is adequate for amplification by PCR. The amplified DNA is used to develop DNA fingerprint, which is used as an important tool in the forensic science. Thus, PCR is very useful for identification of criminals. PCR is also used in the amplification of specific DNA segment to be used in gene therapy. Here, the application of PCR is summed up in molecular identification, molecular archaeology, molecular epidemiology, molecular ecology, DNA fingerprinting, classification of organisms, genotyping, prenatal diagnosis, mutation screening, drug discovery, genetic matching, detection of pathogens. In sequencing, bioinformatics, genomic cloning, and human genome project. In genetic engineering, site-directed mutagenesis and gene expression studies. So today in Applications of Biotechnology Part 3, we discussed about the various molecular diagnostic techniques like the Elisa, PCR and RT-PCR. So thank you. Kindly subscribe, like, share and comment to channel readmedprep Academy. Kindly register for UG and PG NEET type MCQs on our website www.readmedprepacademy.com. Our Facebook ID is readmedprep Academy. Our email is readmedprepacademy@gmail.com. Our Instagram is readmedprep Academy. To join readmedprep Academy, WhatsApp the number given below. Kindly post your questions in the comment box. We will reply with appropriate answers regarding the lecture. Thank you very much.